to start now, right on time, and welcome to this parallel scientific session of International Headache Society Congress. My name is Messoud Ashina. I am from Denmark, and I have a pleasure today to chair this session. And we have some important topics and speakers here. So we're all looking forward for their presentations. We have to be conscious about the time, and the speakers will see the time that they've been allocated to give their presentations. And I will, without any delay, to invite Tayla Rees to the stage. And she is from United Kingdom, basic scientist, and she's going to present it, investigating the role of AMI1 receptor activation and sex hormones in CGRP release and expression in trigeminovascular system. So please, Tayla.
Okay, so thank you very much for the opportunity to present today. My name is Tayla. I'm going to talk about some work that I did as part of the International Headache Society Fellowship at King's College London, working with Dr. Philip Holland and Jan Hoffman. So my disclosures are just this fellowship. So my work focused on menstrually related migraine, which is migraine that occurs in the 2 days prior or post onset of menstruation. And during this time, we have characterized by falling progesterone and estrogen levels, coinciding with an increase in attack frequency. And these attacks are not only more frequent, they are longer, more severe, and resistant to a number of therapeutics, including triptans. So this suggests that the cycling of female sex hormones may modulate some aspect of migraine pathophysiology. However, the exact mechanism is not yet clear. So what we do know is that estrogen and progesterone receptors are expressed throughout the trigeminovascular system, which is the key component in migraine headache. We also know that calcitonin gene-related peptide, or CGRP, is abundant in the system. If you give exogenous estrogen and progesterone to rodents, it modulates CGRP expression levels in sensory neurons and also circulating levels in the plasma. There are reports of changes in CGRP levels across the menstrual cycle in female patients. So this suggests that sex hormones and their cycling could be modulating the CGRP system to drive migraine attacks. However, the CGRP system is a little bit complicated. So the receptor family is comprised of 7 unique receptors, of which multiple are potently activated by CGRP. The most potently activated are the canonical CGRP receptor, comprised of CLR and RAMP1, and the amylin-1 receptor, comprised of CTR and RAMP1. We also know that amylin and the amylin receptors themselves may play a role in migraine pathophysiology, and adrenomedulin is able to induce migraine-like attacks in human provocation studies. So this indicates that all of these receptors may be relevant for migraine, but also possibly menstrually related migraine. So my research aims to investigate female sex hormones and if their cycles are modulating the CGRP peptide and receptor family in the trigeminovascular system. So to do this, I used a rodent model. I used Sprague-Dawley rats, both males and females, to identify any sex differences. And then with the females, I performed vaginal cytology to identify the specific estrus stage that these rats were in. And although the rodent estrus cycle, which is the rodent equivalent of the menstrual cycle, is different from the human menstrual cycle, it does have this reduction in progesterone and estrogen that is similar to what we see in humans. So my research was kind of focusing on estrus following this reduction in sex hormone levels versus the other stages. I focused on 2 parts of the trigeminovascular system, so the peripheral component, the trigeminal ganglia, as well as a portion of the medulla and to the C3 spinal cord, which contained the trigeminal cervical complex. And I wanted to look at if there were any differences in expression of the receptor subunits or peptides in CGRP release, or potentially could there be some differences in signaling between estrus stages for this family of receptors. So first, looking at expression, I dissected the tissues that I previously stated and performed vaginal cytology to identify the estrus stage. I then extracted the RNA from these tissues and generated a cDNA library, and then quantified the mRNA levels using qPCR, with primers designed against both the alpha and beta isoform of CGRP, as well as amylin, and the 5 receptor subunits. So the graph on the right here is a pooled data from males and females, and it's normalized to the housekeeping gene DH. So along the y-axis, as the CT number increases, this means that there is less expression relative to the housekeeping gene. So in the trigeminal ganglia, alpha CGRP is most abundant, and then in terms of peptides, beta CGRP is less abundant, followed by amylin. In terms of the receptor components, RAMP2 and 3 are most highly expressed, followed by RAMP1 and CLR, and then CTR is relatively quite low expression. In the trigeminal cervical complex, a roughly similar pattern of expression for the peptides, with alpha CGRP being most abundant and amylin being least. In terms of the receptors, the 3 ramps and CLR had roughly equal expression, whereas CTR had less expression but relatively more than what you see in the trigeminal ganglia. So this indicates that the mRNA of all 3 peptides and 5 receptor subunits are expressed in these tissues, and all 7 receptors may be present and possibly contributing to trigeminovascular sensitization. So next I wanted to compare expression between the 2 tissues. So this graph on the right shows normalization of the TG data to the TCC data. So interestingly, in the trigeminal ganglia, the peptides were more highly expressed, so alpha and beta CGRP and amylin, whereas in the trigeminal cervical complex, the receptor components were more highly expressed with CLR and CTR. So this indicates we may have some tissue-specific differences in expression. Next, I wanted to compare between the sexes. So this data here is the female data normalized to the male data for each of these tissues. So in the trigeminal ganglia, I saw that the beta CGRP and CTR levels were greater in females. RAMP1 trended higher but was not significant in this particular study. And there was no differences in expression between males and females in the trigeminal cervical complex. So this indicates in this specific study that we had some sex-specific differences in expression. So next, looking at estrus cycle. So here I grouped the female rodents that were in estrus versus the other 3 stages, so diestrus, proestrus, and meestrus, and normalized to those 3 pooled stages. So here this is showing the expression during estrus compared to those in the respective tissues. So in the trigeminal ganglia, I again saw beta CGRP and CTR levels were greater during estrus. Alpha CGRP and RAMP1 also trended higher, but were not significant in this particular study, and this contrasts with a recent publication in mice. And again, no differences in expression between the estrus cycle groupings in the trigeminal cervical complex. So this suggests there is potential for some cycle-specific differences in expression of this peptide and receptor family. So next, I wanted to look at CGRP release. So to do this, you need to have a kit that can actually detect your protein that you're interested in. Michael Garalja, who has a poster here today, recently published a paper comparing CGRP kits and identified the Burton kit, which is able to detect both alpha and beta human and rodent CGRP. So this is the kit that I decided to use. I dissected out the tissues from male and female rats, performing vaginal cytology for the females, and then washed the tissues with synthetic interstitial fluid for 15 minutes, then incubated with synthetic interstitial fluids for 10 minutes, took that solution, that was my baseline amount of CGRP that was being released, and then stimulated these tissues with KCl 60 millimolar for 10 minutes, and this was my KCl stimulated CGRP release. So this is the raw data pulled from males and females. So in the trigeminal ganglia, there is a basal amount of CGRP released, which is significantly increased upon KCl stimulation, and similarly for the trigeminal cervical complex. The values of both the basal and the stimulated CGRP release is roughly equivalent to other studies using this particular kit and looking at these particular tissues. I also normalized the values of the KCl stimulated CGRP release to their respective baseline amounts of CGRP and compared them between the tissues. with the trigeminal ganglia having about a 2.8-fold increase in CGRP release upon stimulation with KCL versus the trigeminal cervical complex, which had a higher amount. But I think this is probably due to differences in the actual tissue size rather than necessarily a mechanistic reason. But what this does suggest is that if this ex vivo model is currently working in my hands. So looking at sex differences in CGRP release, I compared between males and females. For the trigeminal ganglia, there was no difference between male and females for basal release, but the females did have a higher pooled amount of KCL-induced release. There was no differences between basal or KCL-induced CGRP release in the trigeminal cervical complex. When I looked at the normalized data, however, there was no significant difference between males and females. So this potentially suggests that the female rodents in this particular study, their TGs may have been releasing proportionally more CGRP under both the basal and the stimulated conditions. And when this was normalized, that difference in effect was kind of normalized out. So finally, for estrus cycle, again comparing between estrus and the other three cycle stages together. In terms of basal amounts of CGRP release, no differences between estrus and the other three stages, nor males and both TG and the TCC. Similarly, for KCL-induced CGRP release, there was no difference in the pooled amounts of raw CGRP release. When I compared the normalized data, where the KCL-stimulated release was normalized to its own baseline, there was some significant difference between estrus and the other three cycle stages in the trigeminal ganglia. However, whether this is consistent in other studies with other rats or has any practical significance obviously needs to be determined. And again, no real difference in the trigeminal cervical complex. So this indicates there could be some small differences in triggered CGRP release between estrus stages using this model. So finally, I wanted to know about receptor signaling. So the CGRP receptor, upon activation, is internalized into endosomes, whether it is either recycled back to the cell surface or goes through a regulatory pathway. And it has been reported that CGRP receptors can signal from these endosomes, and this may have some relevance for pain. Whereas the amylin-1 receptor does tend to stay at the cell surface and not be internalized. So I wanted to compare if the rat CGRP and amylin-1 receptors were potentially signaling from just the cell surface or multiple endosomes as well. And to do this, I used a technique called bystander BRET, or bioluminescence resonance energy transfer. And essentially what you do is you tag a protein in your compartments with a bioluminescent tag, and then the G protein with a fluorescent tag, in this case GS. And when they come into close proximity, they release light that can be quantified. So if the GS is getting recruited to an area when you've got a CGRP receptor present, then that's suggesting potential recruitment or signaling from the CGRP receptor in that particular compartment. So for the rat CGRP receptor, I saw with the purple line, upon stimulation with CGRP, recruitment to the plasma membrane, which then decreases over time, indicating internalization. And this coincides with an increase of GS recruitment to the early endosomes in blue and the late endosomes in green. In contrast, the rat amylin-1 receptor, upon stimulation with CGRP, we see recruitment to the cell surface as we expect. However, unlike the CGRP receptor, this kind of is just a plateau, indicating no internalization and no increase in recruitment really to the endosomes. So this indicates that these receptors may have different spatiotemporal signaling profiles. So in conclusion, I saw there were some tissue, sex, and estrus cycle stage specific expression patterns in mRNA for the CGRP peptide and receptor family in rodents. There was some small differences in CGRP release between sex and estrus cycle stages. And the rat CGRP and MM1 receptors could have unique spatiotemporal signaling profiles. So future directions, it would be great to be able to determine if the mRNA profiles that I've seen are translated into protein, because mRNA does not always equal protein. Could these signaling profiles be the same in sensory neurons, as I only used a transiently transfected cell line? So whether these differences are present or have any practical significance in our actual tissue of interest still needs to be looked into. And can we potentially target any of these mechanisms to better treat menstrually related migraine? So thank you very much to obviously the International Headache Society for this fellowship and the opportunity to do this research, and Philip Holland's lab and the King's Headache Group. Thank you to Phil, Jan and Eloisa for all your help. My new group at the University of Birmingham and my collaborators at the University of Otago and Debbie Hayes Group. Thank you.
Thank you, Tayla. We have questions? Any questions from audience? So I have just a quick question for you, Tayla, is that if we can translate your research into human clinical world, what would you, let's say, recommend and suggest us to study?
Potentially, if there's more CGRP around in menstruation, you might need higher doses of CGRP targeting drugs potentially, or maybe this increase in beta CGRP means we need more beta CGRP focused drugs that target that more effectively.
Do we know anything about measuring CGRP during the pregnancy?
Ooh, that's a good question, and I think there has been some studies, but I can't remember them off the top of my head, sorry.
Anybody from the audience can comment on that? The CGRP during the pregnancy, anybody knows about that? In fact, that as far as I remember, that was elevated levels, the CGRP during the pregnancy. And what's happening during the pregnancy, the migraine is almost gone.
Yeah.
How can I explain that?
Can't. Maybe in that case it's the lack of the cycling of the sex hormones.
Yeah, it's interesting because you know, we always try to make a relationship between the CGRP and migraine with assumption that it is elevated, but it's not actually the case. You know, just a straightforward case, because we do see that this elevation, and in fact that some of the studies in circulating CGRP suggest also that a bit lower levels, you know, a priori in people with migraine, statistically lower compared to healthy volunteers.
Yeah, I think there's so many mechanisms involved and so many different people. There's got to be, you know, different populations that have different profiles happening. Yeah, yeah.
And it also, the problem was the assay that we used for the measuring of CGRP. There is a variability between the assays.
Absolutely.
And there is a clear need, and you know that you work with Debbie, the clear need for standardized assays, reliable assays at measuring CGRP.
Yeah, absolutely.
Yeah. Well, congratulate with your work, Tayla. Thank you very much for your presentation. Thank you. Good luck. And the next presentation is going to be from Denmark, and Sarah Christensen is going to talk about migraine signaling events in the meninges. Please, Sarah.
Okay. Good afternoon, everyone. Today I will present some preclinical work that I've mostly done as part of my IHS Fellowship Award. And in case you feel something is not clear or missing, please ask questions during the Q&A session or catch me later. So, my disclosures. I'll just start with setting the stage. Ooh. Okay. Yeah, I'll briefly set the stage so you're all aware what's going on in which labs and so. So I was awarded the IHS Basic Science Fellowship Award back in 22 And it allowed me to transition from the Danish Headache Center, where I had worked with Professor Ole since 2012, to move to Boston and work with Dr. Dan Levy at Beth Israel, part of Harvard Medical School. I was there for 20 months, of which 12 months was funded by the IHS. Transitioning with this lot of people was quite an effort, but today I will focus on telling you about the interesting science I did with Dr. Levy. And in the end, I will also briefly touch upon the impact this fellowship has had for my career. So from human headache provocation model, we know that opening of ATP-sensitive potassium channels by the drug lefcromacalin induce migraine attacks. As for CGRP and PACAP, there's some reason to believe that preventing these channels from opening may work as an anti-migraine therapy. KTP channel openers have also been tested in preclinical models, and the collective clinical and preclinical data clearly suggest that the site of action for migraine induction of these potassium channels openers is the meningeal blood vessels rather than a direct activation of trigeminovascular receptors. But nevertheless, there are some major gaps in our understanding of how vascular KTP channel opening leads to headache and migraine, and also how data obtained in rodent models relate to the human data. Therefore, we set out to obtain physiologically comparable data in terms of measuring blood pressure and heart rate in the mouse. And by using in vivo imaging techniques, we obtained information on vascular dynamics and correlated this to grimace assessment as a measurement of pain. We used a standard provocation protocol that was also applied in previous studies and did all the experiments in awake mice. So the lefcromacalin protocol applied was established in our lab in the Danish Headache Center some years back, based on the Pladan nitroglycerin protocol. And we've applied it quite extensively since. The selected dose of lefcromacalin progressively induce cutaneous tactile hypersensitivity when administered repeatedly, so day 1, 3, 5, 7, 9. But a single injection of this dose is in most mice sub-threshold, which you can also see here, day 1, nothing is happening. We also showed that the KATP channel blocker glibenclamide can prevent this induced hypersensitivity. And this was also confirmed in another study from Professor Dusser's lab recently. But for the present experiments, we have focused on the effects day 1 to 5. This seems to be where the most interesting part of the sensitization happens. So I'll dig into the results now. So in collaboration with the telemetric core facility at University of Copenhagen, we compared effects of blood pressure, heart rate, and mouse activity after a single injection of lefcromacalin, so that is day 1 on this figure, but also the repeated protocol days 3 and 5. We found a pronounced cardiovascular effect with an acute drop in blood pressure and a compensatory increase in the heart rate. And the effect size was actually way larger than what was obscene in the human model. However, the magnitude of effect was similar on the different days. So as you see here, day 1, day 3, and day 5, the hemodynamic response was the same. There's a small difference on the heart rate, but it's not significant. Interestingly, this telemetric measurement did not detect any changes in activity levels after lefcromacalin injection. So one thing I want you to note is, on the next slide here, that 2 hours after injection of levetiracetam, blood pressure has returned to normal levels. And this is where we have the peak in the tactile hypersensitivity measurement. Good, so I'll continue with the imaging data. So for this meningeal 2-photon imaging, we place a 3 millimeter glass window in the mouse skull, and through this you can visualize nerve fibers and dura and pia blood vessels. The mice are kept head fixed under the microscope with the ability to run on a wheel. So here we see how a pial vessel dilates in response to levetiracetam, and similarly here in the dural arterioles. And here is summarized data. So on the top row, we have pia responses to levetiracetam, day 1 and day 5. Both days we see a clear significant difference to the controls, but the magnitude of effect was actually smaller on day 5. For dura vessels on the lower row, we see a larger percent-wise dilation than in pia arterioles, but the magnitude of effect was the same on day 1 and day 5. For all measurements, this dilation, it exceeded the 2-hour imaging session, and also exceeding the effect on blood pressure. If you recall from the previous slide, that at 2 hours, blood pressure had returned to normal. So I have 2 key messages from here. So first of all, we don't see this pattern of progressive hypersensitivity to von Frey filaments reflected in the cardiovascular responses. And what is very interesting is this dilation that is actually has a longer duration than the systemic effect of levetiracetam. Good. So we also added the KATP channel blocker glibenclamide, and I will just remind you that this compound was effective on the tactile hypersensitivity measure, but we found here that it did not prevent vascular responses to levetiracetam. Not in the dose we had previously applied in our group, but also not in the dose 25 times higher applied in another study. So you see here that we inject glibenclamide, nothing much happens to the vessels, but then we give levetiracetam and the dural arteries just dilate to a very large degree, and similar to the effect you saw in the previous figure. So, jumping over to the pain assessment by grimace analysis, this is sort of a marker of spontaneous pain in animals. Here we found that levetiracetam induced a small but significant increase in grimace behavior 1 hour after levetiracetam injection, both days 1 and day 5. This effect was not prevented by glibenclamide. So I hope you can follow it on the figure here. We have baseline, and then you see the animals make a larger grimace phase 1 hour after injection, and that is repeated on day 5. But at 2-hour measuring time point, they return to normal. But it's interesting because it's a little different from the hypersensitivity measure that seemed to peak at 2 hours. Good. I will just summarize the key findings from these studies and just interpret a little bit on them as well. A major strength here is that we now have physiological measurements to compare the human and mouse provocation models, despite differences in pharmacokinetic and dynamic differences between the species. All data here is obtained in awake mice, so there's no bias from anesthetics or acute surgeries. So another important finding is that the progressive hypersensitivity we measure with von Frey filaments is not reflected in hemodynamics and grimace behavior. So this thing about a single dose of levetiracetam being sub-threshold is definitely not, it doesn't apply to all measurements, right? Since hemodynamic and grimace appear after a single dose. Then we found that glibenclamide did not prevent left frontocalyp-induced vasodilation and grimace, whereas it worked quite nicely on the hypersensitivity measure. And obviously these discrepancies between the readouts are very annoying because it really disturbs the interpretation of the data. But I think we need to see it in another way, that we can actually learn something about pain perception, allodynia, and sensitivity, that these things are not the same. And this is a long-standing debate in the pain field. How do we detect pain in rodents? And it's clearly supported by the data presented here. My last point is this fact that we have meningeal vasodilation that is lasting longer than the systemic effect on blood pressure and heart rate. And I think that is quite worth noticing. And on that note, I have just a little advertisement for a preprint we put out recently, where we actually have an idea about the mechanism driving this prolonged meningeal vasodilation that seem to be local and not affecting the entire animal. Good. I have a little more to come that I've not presented today. Just a little teaser here. So we have data on mouse locomotion during the imaging sessions. So we can correlate running activity with vessel diameter. And you can see here the trace where the mouse starts running and the PL arteries dilate, whereas the dura arterioles constrict. But what is even more interesting is the calcium signaling in dura nerve fibers that we aim to correlate to left frontocalyp-induced vasodilation, running patterns, and bouts of locomotion as well. I hope that we can see some patterns in the interrelationship between these different factors and possibly learn something about time and space of these different mechanisms in dura mater. For the last bit, just a few reflections on the impact this IHS grant has had on my career. So spending time in Dr. Levy's lab has definitely improved my scientific profile, both in terms of technical skills, network expansion, and collaboration. And collectively, this has enabled me to secure funding and now call myself PI for at least the next 5 years. And I'm very happy to continue our work in Copenhagen with this talented group of people. So with that, I'm at the end. I would like to acknowledge my colleagues and collaborators who have worked on this project with me, especially Dan Levy and Simone in Boston for helping me doing the imaging. And obviously International Headache Society for funding the work, but also a lot of other funding agents have supported the work. And thank you for listening.
Thank you, Sarah, thank you. And it's good to know that the International Headache Society has an impact on people's career. So we have to be...
I'm not too sure about causality, but I hope.
Yeah. I think it's a good message also, to tell other people to become a member of International Headache Society. So please spread the news. So, well, any questions from the audience? Okay, so if I may ask you, is about when you showed the data on the vasodilation, which apparently was a long-lasting, independent from what I saw from the vital signs such as the blood pressure and heart rate. How does it correspond to clinical data in healthy volunteers?
I am not so sure. I find it's when I read the papers, it's not followed as long, right? The traces of hemodynamics.
Yeah, it's in fact that it's quite short lasting. It's not so long lasting. So migraine people, they can behave differently. But if we compare your data from wild-type animals to the wild-type humans. So that will be the best correlation, and that you will see that the head pain they develop, it's a very transient, it's very short-lasting in healthy volunteers, and also the dilation, the magnitude of dilation, it's not so pronounced.
Yeah, and that's, I think it's very important that we now can see that the dose that a lot of people are injecting to the mice based on this von Frey assay of effect is actually physiologically a much larger dose than what is applied in the human model, because the blood pressure becomes way lower. It's like almost like 40% reduction in the acute stage, and it's not that much in the human model. So it's quite clear that we're hitting harder on these mice than what happens in the human experiments. And I think it's important that we know these things, right? Because the mouse is almost like a black box. You just inject something and you measure one thing, but you don't know about all the things you don't measure, right? And they are different to people.
Please. We can't hear you. Maybe can we switch on the microphone? Yeah.
Hi. Okay, much better. So I have maybe a related question. If I recall, in the human models, they don't, when you induce with levkomalkin, triptans don't work in that model. Does that sound right? That sumatriptan was ineffective in those patients? And have you thought about that? And have you ever tried sumatriptan to see if it blocks any of those, either the allodynia or any of your other changes?
Yeah, can you just remind me, was it negative in patients?
So when you refer to human data and sumatriptan intervention, it was in healthy volunteers. Yeah, not in migraine people. Okay. So if you develop migraine attacks after whatever trigger you use, they will report that the usual treatment is effective. Okay, if they take NSAID, it works. If they take triptan, it works. So in this study that you're referring to, that was a study when they studied the ketorolac, NSAID IV formulation, and also compared to the IV sumatriptan. And you can discuss about this assay because it is in the healthy volunteers. So basically in the wild type humans. So it's a different story.
Yeah, we did not test sumatriptan in this live chromogenic model.
No. Do you think it would work?
Yeah, maybe not a full effect, but I think it will work at least partly. I mean, sumatriptan works against nitroglycerin, and there are definitely some shared mechanisms with nitroglycerin and levkomalkin.
Well, thank you.
What was also interesting in your data was about glibenclamide. It was a negative, in fact, that we also see it in humans. Part of the things are, let's say, translational that you see in preclinical studies and clinical studies. But not everything goes, 100% fit to fit.
Yeah, and it's interesting if we ever find out why glibenclamide has worked on allodynia.
Yeah.
Because it seemed to be at least completely different than other measurements, right?
Yeah, but we have to remember that the most of the readouts that used in the preclinical studies was in extracephalic regions. Most of the studies, there's some studies also reported in this, and they said they used the cutaneous tissue, right? Because they stimulated cutaneous receptors. We're not talking about allodynia or kind of allodynia, increased sensitivity directly on the meningeal nociceptors. That could be different. Well, thank you so much, Sarah, and congratulations with your success, and wish you all the best in your future research. Thank you.
Thank you.
And now we are moving to the next part of the session. It's about the Headache Science Award. And the first one I'd like to invite here is Haidar Al-Khazali. He's a physician, he's an MD, a scientist at the Danish Headache Center,
My name is Haidar Al-Khazali. I'm a medical doctor and PhD fellow from the Danish Headache Center. And I would like to state I'm deeply honored to receive the Early Career Award by IHS for my contribution to the field of headache science. This recognition is truly significant for me, and I would like to say thank you to the scientific board for their decision. Thank you. Please just have a brief look at my disclosures. Now, my academic work focuses on migraine, post-traumatic headache, and cluster headache, and has resulted in some publications related to this award. Because we have a little bit of time today, I will focus on 2 of these publications. So, post-traumatic headache and cluster headache are disabling neurological disorders with limited treatment options. This is partly due to our lack of understanding of their underlying pathophysiology. Compelling evidence suggests that KATP channels could be implicated in the genesis of cephalic pain, particularly migraine. Despite clinical differences, post-traumatic headache and cluster headache, they share overlapping features with migraine, raising this important question. Is opening of these KATP channels only implicated in the genesis of migraine pain, or is it a common pathway across different headache disorders? So we raise this question, and I will try to address it by the coming 2 publications. In the first study, we included people with persistent post-traumatic headache attributed to a single mild traumatic brain injury, and these patients were required to report persistent post-traumatic headache for at least 12 months before the study inclusion. We excluded those who had a prior history of any primary headache disorder except for infrequent episodic tension type headache. And in this study, we conducted it in 21 participants. It was a 2-way randomized placebo-controlled crossover study design where they received levochromocalin, a KATP channel opener, or placebo IV over 20 minutes on the first day, and vice versa at the second experimental day. The primary outcome of this study was the difference in the incidence of migraine-like headache attacks during the 12-hour observational period between these 2 experimental days. Our findings, they demonstrate that IV infusion of levochromocalin was able to induce migraine-like headache attacks in 12 out of 21 participants, compared with 3 on the placebo day, so 14%. And the differences are significant. So we conclude by this that administration of IV infusion of levochromocalin was able to elicit migraine-like headache attacks in people with persistent post-traumatic headache. Now, in people with cluster headache, they can present in different stages and different phases. So in this study, we included 10 persons with an active bout phase. We have 15 with remission cluster headache, and we have 17 participants who are in the chronic phase. And they were included in the same study design, so they received either IV infusion of levochromocalin at the first day, placebo at the second experimental day, or vice versa. Now, the difference between this study compared with the other one is that in the other study we had a window of observation which was 12 hours. Here we have, in the primary outcome, 90 minutes. So from the infusion start, within the first 90 minutes, how many developed cluster headache attacks? Of course, we also had data for the 12 hours, but that was not the primary outcome of the study. So here we found out that those who had episodic active bout phase, they developed 6 out of 10 cluster headache attack within the first 90 minutes, compared with 1 participant on the placebo day. In the remission phase, only 1 developed cluster headache attack, and I can tell you one thing, this patient was not in about the prior 3 months, and following this experimental study design with first and second day, he did not go into a bout phase. But the differences are not significant between the active and placebo day. Now in the chronic phase, we have 5 out of 17, so 30% develop cluster headache attacks during the levochromocalin infusion compared with 0 on the placebo day. So we conclude here that opening of these KATP channels can induce cluster headache attacks during the active phases, but not the remission phases. Now, the mechanisms and side of action of how levochromocalin can induce distinct headache types is a matter of ongoing discussion. But we propose some hypotheses. And in one of my first slides in this presentation, I showed you other publications also. So in essence, we have, by CGRP and BiPACAP induced also cluster headache attacks, and also persistent post-traumatic headache patient could also develop migraine-like headache attack. So if we just imagine this is a smooth muscle cell, activation of the G protein coupled receptor or administration of something that can bypass the surface membrane and work within the cell, we can in one way or another upregulate the cyclic AMP pathway. From here, we will activate the protein kinase G, which can actually phosphorylate and activate these KZP channels. On the other hand, we have lipochromocalin, which can bypass all these steps and directly open the same channel. Activation of this channel of a smooth muscle cell will lead to a release of potassium, and from here there will be an activation of the primary afferents within the meninges. It will synthesize and activate these primary afferents and send the information to the second order neuron and third order neuron, and from here the perception of pain will occur, and specifically the headache pain. Now, by administration of different molecules, either molecular triggers or pharmacological triggers, we have been shown to induce different headache types. If you're a person with a history of migraine, you will likely develop a migraine attack. On the other side, if you are a person with cluster headache, you will develop a cluster headache attack and not a migraine attack. If you have a secondary headache disorder, such as post-traumatic headache, you will develop migraine-like features with these exacerbations. So the common understanding from these different results are that the different triggers, they induce different headache phenotypes and different, presentations, which underline that the different triggers are triggers for the generation of headache and not specific for one distinct headache disorder. I would like to thank the participants for sacrificing their time, their, heads, their beings to all these different experimental study designs, and also huge thanks to my mentor, Messoud Ashina, and the colleagues at the Danish Headache Center for all the support and help to get this accomplishment.
This is out of complete clinical ignorance, but is there anything you can give some background about the clinical relationship between the symptoms in post-traumatic headache and migraine? So, because they're getting a migraine attack, is that anything like what they've ever had before? How does it compare to what they have?
Those with persistent post-traumatic headache?
Yeah, that's right, that one.
Yeah. Well, people with persistent post-traumatic headache, they develop after a mild traumatic brain injury, for example, more light sensitivity, more sound sensitivity, and the headache. And this headache is usually almost there every day, and this headache can, within the same day, change in the intensity. So they will get more headache, they will get more sensitivity to light, more sensitivity to sound, and this is why we call it migraine-like feature, because it's a secondary reason, not a primary reason. And we ask the patient when we provoke them with the different substances, is this mimicking your headache exacerbation that you have at home? And they have confirmed this across the different headache studies we have been doing in this population group.
So post-traumatic headache is sort of like a chronic migraine?
Yeah.
Okay.
Yeah,
if not worse.
Yeah, it's
actually.
like, and yes, it is a very interesting question, because what we see, first of all those patients, they develop de novo headache. They never had a history of migraine, okay? This is a mild TBI, which induced persistent headache. According to classification, they fulfill the criteria. But then, this is very typical for the PTH patients, post-traumatic headache patients. They had episodes when you see these waves of, you can call attacks, or attacks-like periods. Could be 1-2 days. Sometimes they try also, physicians also try to give them sumatriptan or other triptans during this day, but they're not quite effective. Or they tried also to give the medications that they usually use for the chronic migraine, but it's not effective either. But phenotype-like, it reminds you migraine because it's photophobia, which is a persistent but exacerbates also during those attacks-like period. So basically, these experimental studies assessed the induction rate of this kind of attacks and showed that there is a difference.
And we have also recently published a paper about the clinical aspect, comparing people with episodic migraine, chronic migraine, and people with persistent post-traumatic headache. And they look very much alike people with chronic migraine.
Oh.
It's published a few months ago in Cephalology.
All right, thank you.
Haidar, thank you so much for your presentation. Congratulations with your award. Thank you. And the next Headache Science Award for IHS goes to Rune Christensen from Denmark. Rune, please come forward. And Rune is also MD and scientist affiliated with Headache Center in Copenhagen, and also Harvard Medical School, Beth Israel Deaconess Medical Center, where he spent some time to learn about preclinical methods and research. And Rune is also quite good in advanced imaging, and he's involved also in multiple studies focusing on pathophysiology of migraine and post-traumatic headache, and published a number of papers. So Rune, we'll look forward for your presentation. Go ahead.
Thank you, Messoud. I would like to thank the IHS.
Can you microphone a little bit
higher? Yeah. I would like to thank the IHS, of course, for this award, and all the people I've worked with throughout the years so far, and hopefully even more so in the future, and of course my supervisor, Messoud, in addition to this. So I'm going to take a slightly different approach than Haidar, in that I'm going to try to briefly summarize some of the key work, at least, that I've done for the past 2 years or so. And it's going to be a bit of an overview, and I hope you will ask relevant questions afterwards if you have any pertinent ones. Okay, so the idea is that I'm going to tell you a little bit about what I think my work has contributed to our understanding of migraine, and especially the attack. And in this regard, it's of course just a single drop of water in the light of all the other research that has also been conducted. So I'm going to begin with the aura phase. As you know, it often precedes the migraine headache itself, and we think it's caused by cortical spreading depression occurring in the brain, observed in animal models, and also in some human studies with neuroimaging. And one of the works I've done has been looking at what is going on in the cortex of these patients who have migraine with aura. And this is a technique we have used that is called quantitative MRI. It's used in multiple sclerosis to look at neuroinflammation. And we try to apply it in migraine as a close collaboration between Basel, Boston, and then the study was conducted in Denmark. And it actually encompassed about 500 participants, 100 of which had migraine with aura. And what we saw in this study was that those participants who had migraine with aura, they had a bilateral increase in a measure called quantitative T2, specifically in the visual cortex. So it sort of corroborates the patient's symptoms of having visual aura and seeing the signal. What does it suggest? Well, it's a measure of water accumulation, and it suggests an edema in this part of the brain, in the visual cortex, that could be related to the aura symptoms or possibly an effect of cortical spreading depression in the brain. Could suggest that there is signs of neuroinflammation in these patients that might also be related to the subsequent development of migraine headache. And I would just like to mention that there are other findings pointing in the same directions. This by Nushin Hajikani, published some years earlier with a specific tracer. So I like to think that this piece of work provides one element bridging some preclinical data suggesting inflammation following CSD to the human experience of migraine. However, there are some nuances to this. For example, we didn't see a relation to the time since last aura in this study. Okay. The next piece of work I'm going to focus on has really been pertaining to what starts the migraine attack. And when we are looking at that in my scenario, I'm going to focus on a peripheral part of the trigeminovascular system. And this is a work that has been done in EMC in the lab of Rami Burstein. And it's using a specialized electrophysiological methods to look at what happens at the meningeal nociceptor. So we know from human neurosurgical studies that the meninges is uniquely pain sensitive, and that these meningeal nociceptors are likely capable of causing headache and even migraine phenotypes in humans. And I conducted 2 studies investigating this. One investigating what PACAP did to these meningeal nociceptors. So we gave an intracarotid infusion, and we continuously recorded the activity of these pain-sensitive nerve fibers that are thought to initiate migraine headache. And here we found that PACAP was capable of activating the nociceptors at a peripheral site, suggesting that this is a likely target for treating and preventing migraine. We also investigated another compound, which we've discussed a bit today, both for Haidar and for Sarah, levcromacalim, an ATP-sensitive potassium channel opener, and what it did to these meningeal nociceptors. And this is very interesting because, as Sarah also mentioned, there is some suggestion that this is maybe not acting at the level of the neurons, but could be acting indirectly through the vasculature. And what we found was that actually, given as an infusion, levcromacalim is capable of activating meningeal nociceptors just as it induces migraine in susceptible individuals. Now, we don't know the exact mechanisms from these studies, but there has been one proposed model which is suggesting that both of them could be acting at the level of the vascular smooth muscle cells, PACAP upstream, and then downstream activating and opening these potassium channels. But my study does not address this, and it's still something that's open for further research, I would say. Okay. Finally, I have also leveraged advanced neuroimaging, not just quantitative MRI, to look at what's going on during the migraine headache itself. So you can imagine we have the nociceptors activating, and now we can look at what's actually happening in the brain. So we might say that something starts in the periphery, but in the end, we cannot have the perception of pain without the brain. Okay. So in this regard, I would like to focus on some work I did using functional MRI, which measures a surrogate of neuronal activity in the brain. And the idea of this study was that, well, we know our patients, they report photophobia during their attack, they seek dark rooms, and in fact, when they're exposed to light or visual stimulation, it can aggravate their migraine headache. So we can use this to look at a neural signature of the regions that are activated during the migraine attack, that is, the regions that are provoked by light. This was a large study. We had about almost 300 individuals with migraine, and importantly, we had more than 100 spontaneous attacks recorded with functional MRI. So a very big sample to investigate what goes on in the brain during the attacks. And then we also had our healthy controls. There were two main findings. One was that in the individuals who had migraine, there was a persistently greater response to visual stimulation in the visual cortex, suggesting a persistent state of visual cortex hyperexcitability. And this was irrespective of the migraine subtype or the presence of migraine headache. The other finding, which I personally find very interesting, is that during the migraine attacks, what happened was that the visual stimulation activated specific pain processing areas, in this case, the anterior cingulate cortex and the insula. Why is this interesting? It is so because we know from simple pain stimulation studies that these regions, they become activated during pain, suggesting an intersection of the visual and nociceptive systems during migraine attacks that are most likely responsible for the perception of photophobia. In further studies, we also looked at the structure of the brain, actually finding that the same regions, the anterior cingulate cortex and the insula, showed structural changes. So these are, of course, persistent changes here with a greater surface area in the cingulate cortex and a lower surface area in the insula. And in fact, the magnitude of these changes were related to disease severity and the chronicity of migraine. So in short, what this has told us about the phases of migraine is that there seems to be neuroinflammatory signals related to aura. Meningeal nociceptors could have a key role in initiating the migraine headache, as shown with PACAP and levcromacalim as a likely mechanism of action outside the brain. That pain areas become hyper-responsive during the attacks, and specifically during visual stimulation, and that structural changes in these same areas seem to characterize the migraine brain. Finally, I would just mention that we actually conducted, in close collaboration with Haidar as well, studies between migraine and post-traumatic headache to see if maybe similar changes were occurring in these patients who have a very, very similar phenotype in the clinic. And we found some of the same things going on. In post-traumatic headache, those patients who reported photophobia during the scan, they exhibited the exact same patterns of hyperexcitability, again pertaining to the insula and the anterior cingulate cortex. Looking at structural brain changes, we also found overlaps, not the exact same areas. The anterior cingulate cortex was also altered, but in addition, the post-central in post-traumatic headache. However, there were also differences. When we looked at the functional MRI response, this persistent state of hyperexcitability we only found in the individuals with migraine. We did, however, have only 100 participants with post-traumatic headache, so a lower sample size. In addition, we found white matter changes in tracts that run to the somatosensory cortex in post-traumatic headache, though these weren't present in migraine. So there seems to be some similarities, but also some key differences in this regard. The key messages: migraine with aura is linked to neuroinflammatory signs. Häckert and chromaculum likely triggers migraine by activating the meningeal receptors. And both migraine and post-traumatic headache are linked to partly overlapping dynamic and persistent alterations in similar pain processing regions. Lastly, I would like to thank again Messoud, my other supervisor Faisal, all my colleagues at the Danish Headache Center, Ami Burstein and his lab, and Professor Hachikani and Grantier, all research participants, and especially the Lundbeck Foundation for their support of many of these studies. Thank you all for listening.
Thank you, Rune. Thank you for an excellent presentation. Unfortunately, we don't have time for questions. I'm sure that somebody will approach you and ask you some questions. Thank you very much. And I would like to congratulate again all the speakers for their excellent talk, but also their scientific achievements, and I wish them good luck. Thank you.